Hi Higher, so we are looking at chromosome structure mutation and also how that can be important in evolution in the DNA and the genome unit. Okay, so we've already looked at changes in genes, so specific genes inside them, whether their base sequence is the same, whether that's a substitution, an insertion or a deletion, you have changed the bases. But there's a bigger picture here, which is basically at the level of the entire chromosome. So involving entire genes, not just the sequence, but moving entire genes. And that could be something that changes the number of genes, it could change the order of the genes, it could change what chromosome they're on. They are quite often lethal because they're quite substantial changes. These ones, we're looking for deletion, inversion, duplication, and translocation. You have to find a way to remember that one, as well as your CID from your point mutations. So deletion is again kind of obvious. We are just going to delete. So here's a section of chromosome and here are your genes labelled A, B, C, D, E, F all the way along. And deletion would mean that I would take something out and I would get a shortened chromosome. Now that's obviously going to have a fairly major impact. Inversion hopefully also sounds okay, because if you invert something you just flip it over. So what you need to do here is to take out 2 or 3 or more genes, flip them over and return them. So I can take something that reads A, B, C, D, E, F, etc., flip it over and I get A, B, C, F, E, D, G, H, I, J. Okay, so we've not changed the number of genes here, but we've changed the order of genes, and that can be super important, especially if you swapped her out where its promoters were and things like that. Duplication, now you have to be very careful on the definition here. So you're obviously making a copy, okay, because you're duplicating the gene. But the way you're doing that is you're taking from the homologous chromosome. So if this is, say, chromosome 1, then you would also have another chromosome 1, because you got one from your mum, one from your dad. So to get a duplication event, what happens is you splice in some from one into the other. So in this case, we have copied the B, C, D, E section, and you've now got 2 copies of 4 genes on just one chromosome, which means if that then goes into being in a particular sex cell, then when the new homologous chromosome comes along, you're going to end up with 3 copies of the same gene. And lastly, we've got translocation. And hopefully you can see where this one's going in terms of the word. So trans is basically moving something and location would be to a different place. So what we're doing is we are shifting one chromosome onto another chromosome. Now, because it's not going to be the same homologous chromosome, then it won't come up read as a duplication. So here's an example, just another one of the ABCs. Okay, let's assume we have a totally different chromosome, which is running with the Greek letters to make it obvious it's somewhere else. And here would be after a translocation event. So what we've got is now reading our ABC whatever, and then our Greek letters at the end of that. So what's happening there is we end up with a longer chromosome, but genes on it that were not normally there. So all of that is just a straight, you've got to know each of those processes and recognize them. But what you also have to recognize is why they are important, because mutation is the raw material. It gives you the potential for selection. We get some variation from things like meiosis, which is the process of making gametes, the fact that we have in sexual reproduction random fertilization. All of that helps to just mix about all the alleles that are already present. But if you want any new alleles, then it's from mutation. And that will allow you to have differences of advantage, to have some members of the species which are actually more likely to survive and therefore pass on their genetics. And over many, many, many generations, that potentially leads to evolution. Okay, so there is one specific that you're supposed to recognize for genes, which is that duplication is a really, really important thing for evolution. If you've got duplication, it means that you have 3 copies of a gene. Now, if one of them is mutated, you still have 2 which are doing the normal thing. So you would still get the original protein, but now you've also got potentially a different protein, and that might give you selective advantage, which means that you would then keep it, or it might not work at all, but then you've still got two that are working. It's a kind of belt and braces approach to the whole thing. It means that new variation could still be tested while you have a functional system as a kind of safety net. And that's the end of that key area.